Okay, so there's audio. You don't know? Okay, so I still can see my basket. The last two things I want to show you from this database are fairly simple. One, I'm going to go back to the usual suspect, hemoglobin alpha subunit from Homo sapiens. If you need the code, does anybody need the code?
Sorry?
Do you need the code, or do you still have it in your basket? Yeah. So then I'll give you the code. P69905. P as in Peter, 9 as in 9. 9 again, 05.
999.
Those are 3, and then in the meme it's like 4 times, and banging the table. Alpha subunit from hemoglobin. What's that?
No, I deleted my basket, that's why I didn't.
Well, you have the code. You can find it easily. And since you can do it from Google, that should be speedy. So here in Homo sapiens, the two things I want to show you, the first one is simple. Go to sequence again. And here we already did the blast. But if you click on that drop-down menu, you can find a couple of other things. One or several are very similar. Protein parameters. Hang on. One. No. That's better. So right here. First of all, the length I already mentioned, the mass of the subunit, and then prot param, prot scale, compute, PI, molecular weight are very similar. So select, let's go for compute, PI, and molecular weight. That one, what it's going to try to do is taking into account the number of amino acids with protonated sites or protonatable sites, acids or bases, it's going to estimate at what pH the protein is going to be neutral. And the other one is the molecular weight, which we already have here based on the mass. So this one is very weird because it doesn't give you straight up the information, but what it does is divide the proteins on the secondary structures, if you remember from biochemistry. If not, look in your book, or the actual peptidic chain. And all of these, they are in color because you can click on them. So if you want to find out the isoelectric point of this whole protein, you will click here. If you would like to define another region that is not listed here, you can use these numbers. For example, I could set not something between 22 and 38, let's say 37. I want to know the PI and molecular weight between 37 and 95 that are not listed here. So you could do that here. For now, I'm going to click on this one, the whole chain. And here we have the sequence again. Just it's verifying with us that we got what we were looking for, not only by numbers, but by the sequence, the molecular weight. And this is going to be something that experimentally you can measure in a protein electrophoresis, and a theoretical isoelectric point. So later in this month, when we review, or in the next month when we review isoelectrofocusing and electrophoresis, this information is going to be relevant because you already saw that alpha, beta subunits, or from Homo sapiens and other proteins from other animals, they are very similar. But they do have mutations, and those mutations are going to affect both the mass as well as the isoelectric point. So those are going to be tools, these are going to be theoretical tools for experimentation if you want to separate those samples. Or identify. I don't want to be morbid, but if you find blood in the jungle, biologist, and you want to find out if that was for a human, a chimpanzee, an orangutan, this will be crucial because you can identify hemoglobin in that blood and, with a little bit of luck, if these properties are different enough, distinguish the source. There's another set of properties here. I'm not going to cover them. Some might be useful, some might not be useful. There are a few like here in prot param. Let me see if this one. there's other properties that you can try to estimate from the sequence. One of them is the absorbance under UV light. So here we have that one, or at least we should have it. Yeah, down here. I know that I haven't covered this a lot in my classes. I don't know if there's instrumental chemistry or something like that where you cover this. But here, this is the experimental property that you could measure. If you were to have 1 gram of alpha hemoglobin from human, and you put that in a solution and take to the spectrophotometer, it should absorb this much at 280 nanometers of wavelength. This is a prediction based on the sequence. So if you do the experiment for real, it might be off, but it gives you a good guideline if it's going to be something that absorbs UV light or not. You can calculate this by hand, but this program, this part of the database does it for you. Since we are not going to do experiments with UV light, well, this is not going to be as useful in the class, but it's something that can be important or interesting. And the last one, the last property, the last piece of information that I wanted to show you is interaction. An interaction, does it look like this in your computers? How does it look? Like a matrix, right? A column and a part on top.
Yeah.
Darn it. Do I have Firefox? Oh, let's see if I can open it there. So how are my levels? Good audio? Thank you. Oh, what? Did I click on something?
Firefox.
No, but back here. Do you see that animation?
No.
This animation is trying to tell us several things. This has become very common. What's that?
Where is that the other picture?
Yeah, that's a very good question. I click accidentally on it. So let me go back. And let me go to interaction. So I must have clicked here.
No.
Or here. Where did I click? Go back in the stream and tell me.
Oh, yes.
Okay, it was this one. Did you see which it was? The complex portal. So you have two sections. I'm gonna go back so you can see it. You have two sections under interactions. First, the part that doesn't look okay in my computer. Then BioGrid, and then complex portal. Click on the first entry on the complex portal, which is what I did accidentally. But I didn't want to go there, but well, since you already saw it, maybe you can make better sense of this than me. We have this symbology. Don't look at this too much for now, but here we are looking at the known interactions of this protein. The obvious for an alpha subunit of hemoglobin is the beta subunit of hemoglobin. So here we have alpha and beta. Yeah, it says unspecified role, but we do know the role, right? It's binding oxygen. And that leads us to the next interaction, the cofactor, the heme group. This is very simple for this protein because it's telling us you're going to have 1 and 2 beta subunits in a complex, 1 and 2 alpha subunits, and each one of those proteins is going to have a heme group. This is the functional complex of hemoglobin. If you remember from biochemistry, 2 alpha subunits, 2 beta subunits, and they have the heme group. So this is a graphical representation. This particular one, we are lucky because the interactions are simple, but in general they are not like that. Either this information is empty and nobody knows what it interacts with, or the interactions are with everything and they are hard to read. That's why I didn't want to click on this one, but this one worked out rather nice. Let me try to copy the UniProt into Firefox. Come on, collaborate. Oh, my setup is working against me. Pay no attention to that part, it's just my own preference for encrypting some of my connections to the internet. Here we go. So this is what I really wanted to show you. Here in this table, what we have is a matrix of several proteins, starting with hemoglobin alpha, and then following with other proteins of human, particularly beta, hemoglobin, and epsilon. And what this table is telling us with these colors is that human, or whichever, what it's telling us is that alpha, hemoglobin from human, interacts with all of these proteins according to the color gradient. So the most important interaction is with beta hemoglobin. But it's known to interact with all of these other proteins. Of course, this is more complex to follow, right? And you have to find out what these other proteins are to make a little bit more sense of it. Whereas this complex portal, actually I haven't clicked on that before, but it looks very nice because it gives you a relevance in the biological sense. What other interactions it requires to be assembled as a functional property, protein. Okay, so I guess that's about it. Should I use Firefox for the remainder of the class? I guess so, right? Questions? You sure? So the purpose of this section is, in my opinion, to look at a very rich database of proteins. That is a crucial part of this. There's only sequences for proteins, no sequences for DNA, no sequences for RNA, no tRNA or ribosomal RNA, only proteins. So we are looking specifically at the products of some genes, which implies that some of these sequences and in some of these data, in this database, you should be able to find where there exists information for that. Splicing, alternative splicing proteins. In the case of hemoglobin, we didn't see any. But if you look for more complex functions in cell biology, you're likely to find 1, 2, 3 splicing variants. So I'm not covering that, but you should be aware that they exist. And they will be listed here. In this case, I think we don't have anything other than natural variants. Now, you can erase your basket, but I recommend you save this for a little while. At least the code, okay? Now, for a totally different database, I wanted to talk about OMIM. And there's a presentation on the website, on the portfolios. I recommend you take a look at it. Because even though that very same information is over here, in the presentation I have abstracted a couple of items that are very important. In fact, maybe I should open it here. Where is my Safari? Here we go. Let's see, is that number 3 or number 4? No, number 4? 5? Whoa, we have covered a lot of things. Okay, so for that one, let me see quickly here. Yeah, I'm going to start at the end of the presentation. Here. Sorry to take it in a very different way than it's written, but there is something very important to understand from this database. Even though it's named the online Mendelian inherited what? OMIM. Hey, where? Here. Online Mendelian inheritance in man, not in human. This is very sexist. It actually makes a distinction between this basic classic Mendelian inheritance. What do you remember about that inheritance? What is it supposed to mean? That is what, well, it implies sexual reproduction, for one. For the second, Mendelian inheritance also almost imply that there's one gene and one function, one genotype and one phenotype, right? That's the paradigm. In fact, in some of my classes in molecular biology, cell biology, I show you the molecular biology central dogma, DNA to RNA and to protein, which implies one gene, one RNA, one protein, and implies one function. I want to show you this table because here, even though the database is Mendelian inheritance, if you look at the phenotypes, okay, we have about 6,000. There's about 4,000 genes that are known to cause mutations, but there's also statistics for this. Number of genes with only one phenotype, it's actually less than a third of the total number of genes in the database. So what this table is saying very calmly, without much noise, is there's no only Mendelian inheritance here. There's non-Mendelian inheritance too. And in reality, it's non-Mendelian. There's going to be many phenotypes that either have two genes. Sorry, there's going to be genes with two phenotypes, genes with three phenotypes, genes with four phenotypes. The reason for that is not obvious, but if you look at different genes, you realize that they are interconnected in terms of function. One gene usually doesn't have a function in general. There's also, not here, let's see, and that is what I'm looking for, but I cannot find it here. Very interesting description. So we have several pieces of information: gene description, gene and phenotype, gene description and molecular basis, phenotype description of locus and molecular basis, and other, mainly phenotypes with suspected Mendelian bases. So here the curation of this database all revolves around the phenotypes and a little bit of molecular biology. But if you look over here, we have the classic Mendelian inheritance, either autosomal, linked to the X chromosome, to the Y chromosome, so maternally, parentally inherited or paternally inherited, and mitochondria. Is mitochondria Mendelian?
No.
No.
Technically it's maternal, but it's not linked to the X chromosome. But there are some mutations with known phenotypes, with known diseases, and they are collected here. Even though they are not part really of our genome, and they are not inherited in a Mendelian fashion. Although there's a couple of reports to the contrary. There seem to be some exceptions. Now look at these icons that are very simple. When there's only going to be gene description for something, there's going to be an asterisk. When there's going to be more information, there's going to be a plus sign. There's going to be a hashtag, if you will, which is not the original name, and a percentage. And these codes here, you're going to be able to find in the database. So let's jump into the actual practice of what's going on here. Go into the database and look for, yes, there's also this obvious aspect. It's inheritance in man or in human. So here this database is only going to be about humans, about Homo sapiens. No chimp, except we're going to see in a very special instance. So type sickle cell anemia into your browser. Sickle cell anemia, like that.
But do you need A at the end?
Yeah, but helpfully autocorrect can do that for me. So
Okay.
you want me to waste the time of another letter? There it goes. And as you click look for the search, this is what you get. Now remember the codes? What does the hashtag stands for?
Trends. No, no es cierto.
Not always.
Phenotype.
Phenotype description, so how does the disease look like, and the molecular basis is known. So in a way it's saying we know the function and why the gene affects that function in the sequence, or because of the sequence. But not only that, there's the asterisk. What is the asterisk? There's a description for the gene. So it kind of goes together with the molecular basis, right? We know the phenotype, but also know the gene. The same for this one, fetal hemoglobin quantitative trait. You can always associate a thalassemia, which is pretty much another way of saying anemia, with this with mutations in these proteins in hemoglobin, fetal hemoglobin, gamma hemoglobin. Remember what this percentage stands for? The percentage stands for phenotype description, how it looks like, where in the chromosomes there is a mutation or some change associated with this phenotype, and the specific molecular basis is not known. If there's a mutation in the gene, or if there's a mutation in the regulatory elements, it's not known. It's just known that something happens in that particular chromosome near or at a very specific gene that causes a phenotype. So it really looks more like a clinic something, right? Because you need to correlate this information from the molecular level with the organism level. But the organization actually facilitates that. If you are looking for this plus, you know what to look for here. There's a plus. Doesn't seem like there is any. So if you were looking for that set of information, well, there's none, right? Asterisk, asterisk, hashtags, percentage.
Yeah, there's one.
Where is it? Which?
Number 21.
21. Ah, you click fast. All the training is paying off.
All the games.
I didn't say that. What? Is there any question? No? You were just choking?
No, I was just like, oh wow, I found it.
Oh, okay. More power to you.
Yeah.
Which is actually, look, the sickle cell anemia, it's related somehow to this high density lipoprotein, the cholesterol levels and whatnot. And here are the matching terms, sickle cell anemia. So it's a strong match. But if we were only looking for mutations in the hemoglobin that affected that under that plus particular description, clearly we didn't find any directly. That doesn't mean the information is not there. It's just that it's not classified only as that type of category. So I'm going back to the first one, the first page of results, because with phenotype description and molecular basis known, which sounds pretty interesting to me, I find the definition for the sickle cell anemia. And even though this looks like Google maybe 10 years ago, it actually contains far more information than Google did or ever did. First, a code. Each one of these codes, it's like the one in UniProt. If you ever need to go back to sickle cell anemia, you just need that code. I haven't tried it in Google again, but it's likely that it will work there because it's very unique. Second, it identifies whatever is happening in this disease with this cytogenetic location. What is a do you know what a cytogenetic location is? No? Well, it's like a map. It tells you it's either north, south, east, or west, and in which city. So here we have 11, which stands for chromosome 11. P or Q, depending on the short arms or the long arms. And a position relative to the centromer of the chromosome. So if you were to design, let's imagine you want to try to design a test, a genetic test for that particular disease. You want to do it in newlyborns, and you need to locate that via a test. Well, if you can somehow design an antibody, a stain, any test that separates that chromosome and can check what's going on there, there you go. You could measure it and determine if that disease is present in that sample. Yeah, I think that's about it in that one. So I'm going to click. Did I click? There we go. Yes. Now the outline here is not as colorful as UniProt, but it's also as useful as UniProt. On this side, we have, technically, this is like the index for this page alone. We have phenotype-gene relationship, clinical synopsis, which kind of is like a description of the disease in the organism level. Text with description, clinical features, diagnosis, clinical management, pathogenesis, molecular genetics, population genetics, animal models. Remember that I said this was about humans, except in the animal models. If there's an animal model, mouse, rat, guinea pig, maybe even Xenorhabditis elegans, to study this disease, it's going to be listed there. Of course, just as in UniProt, there's going to be diseases that have no information whatsoever for many reasons. But you are going to be entries that lack all of this, or some of it. There's history, reference, contributors, and of course, as in any database, when was it created and when was it this last updated. And the way this is depicted here, it's sometimes really boring. It's mostly text, and so you want the information, you have to read. There's no nice tables or nice graphics, but the information is still here. So first, here, the phenotype-gene relationship. The location again, cytogenetics, a phenotype a number, which corresponds to the number we find in the search. What do you think AR stands for in inheritance? Look at this feature of the web page that I like a lot. If you click on it, it's going to tell you it's autosomal recessive. So what does that mean? That in order for that disease to manifest, you have to have two recessive genes. But at the population genetics levels, you're going to have individuals with the correct dominant and the recessive hidden. Right? And if we were like yeast, there will be two with the recessive, with one recessive, one with two recessive, and one with two dominants. But I think families with 4 kids are unusual nowadays. Phenotype mapping key. This is useful also within the database. To remember the correlation of this hashtag, the molecular basis of the disorder is known. And the gene locus, specifically not the cytogenetic location, but the gene location in terms of the sequence. Where does it start and where does it end? In the text, it's actually pretty self-explanatory. The number sign is used with this entry because sickle cell anemia is the result of a mutant beta globin, this gene, in which the mutation causes sickling of hemoglobin. You know what sickling means in this context? That these are normal red blood cells, these are sickle cell anemia blood cells. As far as I know, nobody here has ever shown that in my experimental biology lab, and we have looked at many blood samples. So, where is it prevalent? And let's see if the text says that. Clinical features. So, if we were to look at where this disease is prevalent, or this mutation is prevalent, the population genetics section should tell us, right? And what does it say? It actually says Sub-Saharan Africa. What does it mean? Well, actually below Egypt, below, what is it there? My geography of Africa is not really good, but it's below that. What's that? Morocco? Morocco is actually way north, yes, but that too. But there's other 2 or 3 countries at least under.
The Congo.
That is way down, but yeah, it's included. Hemoglobinopathies, a fancy name for diseases related to hemoglobin, occur at particularly high frequencies. So sickle cell anemia, alpha plus thalassemia. And the reason the population genetic is individually, each one is protective against severe Plasmodium falciparum malaria. So if you were to draw a map of this mutation versus a map where Plasmodium malaria is prevalent, they will overlap. The pressure of selection of the prevalence of the disease versus the prevalence of humans has made this mutation to be very common in that region. It could happen anywhere, but it's favored in that environment. So this population genetics, as you can see from the beginning, goes into a lot of detail into the population of humans with these mutations, if there's information. Hemoglobin has been studied for ages, so there's a lot of information here. It's pretty exciting, right? If there's enough information of a disease, if you ever want to learn from the biochemical chemistry point of view, this is the place to look at. But there's something for biologists too. The animal models, for example. There's a mouse human hybrid cell. Have you ever heard of those? No, those are pretty exciting. And if there's time, maybe we should talk about those here. But in cellular biology, wow, we didn't cover that chapter, did we? No, we did actually. I'll bring it up later, don't worry. There's transgenic models and other knockouts in mice, many techniques to study these diseases. And because these are based on the literature, anything that's been published related to this disease is going to be annotated here. Not everything, but the most relevant under the context of sickle cell anemia. What about, would you like to read diagnosis? But can you take a guess at what the diagnosis will be? Well, we're going to have two. We're going to have two. If you have dominant recessive, that's going to be one diagnosis. And that's probably going to be through a blood sample. But what about the double recessive? What would you expect with somebody with anemia?
Genetic.
No, they're going to be like, and I don't want to insult anybody, very fair skin, or even if dark skin, with a pale complexion, right? The implications are obvious. If you are anemic, you're not going to be 100% strong. And that's what we're going to find. We are supposed to find those two things. Oh wow, this goes really into detail. This one, for example, because the molecular basis is known, it's even recommending PCR amplification. That's fancy, right? If you're in the sub-Saharan desert, I'm not entirely sure you can pull it off. Not for a large number of individuals, for a few. Let's see. Wow, this has become extremely high-end. Well, look, it goes into the early pregnancy diagnosis. Do you think there's actually clinical management for this?
Yes, there is.
I don't know. Let's find out. Well, there's agents, I didn't know that increase fetal hemoglobin production. Hydroxycarbamide, decitabine, and butyrate. So that makes mosquitoes like you a lot, huh? So for children with the disease. Stimulating fetal hemoglobin by increasing gamma globin synthesis in patients with sickle cell anemia would be expected. Azacitidine and hydroxyurea. Okay, so there's treatment. But what do you think will happen under treatment to the resistance or the tolerance to malaria? Right? It would be counterintuitive. What about using CRISPR?
We can have all in this one.
What's that?
We can have all in this one.
Either you stink or you die of anemia? You know why I say either you stink? How is urea processed and excreted? It's through the urine, and it's the fish-like smell. That's nitrogen. So if you are consuming hydroxyurea or butyrate is that like cheese going bad smell. So they are kind of not the greatest smell in the world. But hey, if you are anemic and you really need it, there's no other way.
Like, this is like a hereditary symptom. Syndrome. I think that there isn't a cure, it's just like a treatment.
Yeah, that's why it's called clinical management.
Oh, okay.
Right? It's not the cure or treatment.
Yeah.
It's management. It's ameliorate the symptoms in the lingo of the medical doctor.
Si solo tienes una copia, se supone que a veces ni te das cuenta, ¿no?
Right. It will be Well, so since you asked that question, let's emphasize the part of Mendelian inheritance. If this disease, that mutation, that phenotype is actually Mendelian, that will happen. That is, you need to have 2 recessive copies for the disease to manifest. But there's 2 instances where that is false. One of them, well, the obvious, if it's not Mendelian. But have you taken human genetics or human
We're taking
inheritance?
it right now.
You are taking it right now? Well, they should teach you this concept of, I think it's called penetrance. So instead of having, let's say, 2 dominants and 2 recessive and then the middle, most non-Mendelian inheritance, instead of you being located here, here, or here, there's actually a gradient. And if there's a cell where there's a richness of this protein, of the recessive one, you might start manifesting the disease. But if your cell types, let's say the liver, are mostly, even if you have this combination, mostly producing the dominant, nothing is going to happen. I don't want to go into detail if you're taking that classes, but particularly in females, sometimes there's different cells with different genomes. And you can have mixtures of different X's from your fathers or from your mothers in different cells in your body. And these are called mosaicicity. And of course, during mosaicicity, there's no Mendelian inheritance. So there could be, and this is totally weird, there's cases of people that have an immunodeficiency, but only with a specific organ in their bodies. No, that's generally, I haven't seen it like that, but maybe the severity. As far as I know, if there's mosaicicity, it's like low. Well, I'm not a human geneticist, so that's why I don't want to go into detail. But those things can happen. So even if you have dominant or recessive, accidents of cytogenetics might lead you to manifest the disease because it's not Mendelian. That's the only point I want to make. If it's not Mendelian, that happens with mitochondria. Mitochondria are not exactly the same across your body. There's parts of your body that could accumulate mutations in the mitochondria. You lose it, and you only go blind, but you can live a normal life, so to speak. But that happens because they are non-Mendelian. What? Oh yeah, so it's time. I always get carried away, but it's fun. It's nice of you to ask questions because then we can explore the limits of this information. Here, in a way, because lots of information are known and it's already assigned as autosomal recessive, it would be unlikely to find this disease manifesting as a gradient with different penetrancy. But that is not going to be true for every single disease. So next class we'll continue not only with some of the other features in the database, but also with the clinical resources that are over here. For example, clinical trials. Okay? Well, guys, have a nice weekend.